So, me caveman, let's go. Number 10. The Xiahe Mandible. In 1980, a Buddhist monk was praying inside a cold limestone cave in Xiahe County in the Gansu province of China when he found half of a human jaw pressed into the rock. He did not know he was holding a member of a lost species. The jaw passed from the monk to the 6th Gungthang living Buddha, and then to Lanzhou University, where it sat in a drawer for decades. Nobody realized what it was. The cave, called Baishia Karst Cave, sits 3,280 meters above sea level on the northeastern edge of the Tibetan Plateau. That is higher than almost any ski resort on Earth. The air there is thin, the oxygen scarce, the cold brutal. And yet something human, or almost human, was living there in the deep past. The specimen is a robust right half of a lower jaw. It still holds 2 large molars, and it lacks a chin. Those are archaic features, the marks of a hominin that was not one of us. Researchers dated the crust of carbonate stuck to the bone using uranium series dating, and the answer stunned them. The jaw was at least 160,000 years old. Now consider the problem. No DNA had survived in the bone. That is where the story becomes something new. A team led by the Lanzhou University geologist Chen Fahu, working with Jean-Jacques Hublin, Frido Welker, and Dongju Zhang, did not read genes at all. They read proteins. From the dentin inside a single molar, they extracted ancient collagen and analyzed its amino acid sequences, a young technique called paleoproteomics. The proteins carried a signature. That signature matched the Denisovans. Until that moment, Denisovans were known from exactly one place, a single cave in Siberia, from a fragment of a finger bone. The Xiahe jaw sat more than 2,000 kilometers away. It was the first Denisovan ever found outside Denisova Cave. In one stroke, the range of this ghost species exploded across Asia. The work was published in the journal Nature in 2019. There is a deeper implication, and it is the reason this jaw makes people uneasy. It means Denisovans had adapted to extreme high altitude more than 100,000 years before modern humans ever set foot on the plateau. Later work confirmed the picture. In 2020, researchers recovered Denisovan mitochondrial DNA from the cave's sediment. In 2024, a new study, led by Dongju Zhang and Huan Xia, argued Denisovans occupied the cave from roughly 200,000 to 40,000 years ago. The animal bones told the rest of the story. There were cut marks and butchery traces on blue sheep, on wild yak, on gazelles, and even on birds and carnivores. These were not passing visitors. They were residents of the high plateau, hunting and processing prey across tens of thousands of years, in a place where the air itself fights the lungs. But listen closely, because here is where the controversy lives. The identification rests on proteins, not on DNA. Some researchers argue that reading a handful of amino acid positions cannot fully rule out an unknown archaic population. The mainstream view says this is a genuine Denisovan, proof of a pan-Asian range. A more cautious view says it may be a distinct regional group. And a fringe view whispers that we do not truly know what this was at all. Which raises a harder question. What happens when the DNA is intact, and it tells you that a person's own great-grandparent was a Neanderthal? Number 9. Peştera Oase. In 2002, cavers exploring a flooded cave system in the Carpathian Mountains of southwestern Romania found a scatter of bones. The place was named Peştera Oase, which means the cave with bones. Among the remains was a single human lower jaw, later labeled Oase 1. Radiocarbon dating placed it between 37,000 942,000 years old. That makes its owner one of the earliest modern humans known from Europe. When the genome was sequenced and published in Nature in 2015 by Xiahe Fu, David Reich, and Svante Pääbo, the numbers did not look real. This individual carried between 6 and 9% Neanderthal ancestry. That is more Neanderthal DNA than any other modern human ever sequenced, then or since. Most living people outside Africa carry only about 2%. The genetics went further. 3 chromosomal segments of Neanderthal ancestry were enormous, each longer than 50 centimorgans. That length matters because there is a molecular clock hidden in it. Every generation, the process of recombination chops inherited DNA into shorter and shorter pieces. Long, unbroken Neanderthal segments mean the mixing was recent. In this case, the segments were so long that the man had a Neanderthal ancestor only 4 to 6 generations back. A great-great-grandparent, or perhaps a great-great-great-grandparent, was a Neanderthal. This is the most direct genetic evidence ever found of a face-to-face encounter between the two kinds of human. Not a distant statistical echo, but a family tree with a Neanderthal branch just a few rungs up. And yet, here's the twist that keeps this find quiet. Oase 1 shares no more DNA with later Europeans than he does with East Asians. His population left essentially no genetic trace in anyone alive today. The interpretation is sobering. He belonged to an early pioneer wave of modern humans that pushed into Europe, interbred with the Neanderthals already living there, and then died out. His high Neanderthal fraction is not the source of the Neanderthal DNA that modern Europeans carry. That came from a different, earlier event. His whole line simply ended. The DNA itself was degraded and contaminated, and the researchers had to filter out modern human contamination with great care. The jaw's shape is a mosaic, mostly modern, but with archaic large molars, which had puzzled anatomists before the DNA settled the argument. The mainstream view calls Oase 1 a doomed pioneer, genetically swamped. Archaeologists see a mosaic frontier where species mingled at the edges, and the more speculative view suspects that many more failed hybrid populations are still waiting to be found. So what happens when that same pattern, a recent Neanderthal ancestor, shows up not in one person, but in a whole group? Number 8. The Bacho Kiro Cave Genomes. Deep in Bacho Kiro Cave, near the town of Dryanovo in Bulgaria, archaeologists found human remains directly dated to about 45,000 years ago. These are the oldest, securely dated Upper Paleolithic modern humans in all of Europe. They were found alongside a rich toolkit, stone blades of the initial Upper Paleolithic, bone points, pendants, and ornaments carved from cave bear teeth. For years, scientists argued about who made those tools. Were they Neanderthals, or were they modern humans? The bones were too fragmentary to be sure. Some fragments were so small they were first identified as human only by screening their proteins. Then the genetics arrived and settled it. A team led by Mateja Hajdinjak and Matthias Meyer in the lab of Svante Pääbo at the Max Planck Institute sequenced genome-wide data from at least 3 individuals dated between about 45,900 and 42,500 years ago. Published in Nature in 2021, the answer was clear. These were Homo sapiens, but the shock was in their ancestry. All 3 of these earliest Europeans had Neanderthal ancestors only about 6 generations back in their family trees. Their Neanderthal ancestry fraction sat around 3 to nearly 4%, higher than that of most people alive today. 6 generations is roughly the distance between you and a great-great-great-grandparent. These were modern humans whose recent relatives had been Neanderthals. And there is the familiar, uncomfortable ending. Genetically, the Bacho Kiro people were more closely related to later East Asians and Native Americans than to later Europeans. They contributed little or nothing to the modern population of Europe. A later individual from the very same cave, about 35,000 years old, belonged to a completely different genetic group. Even at one cave, one people had replaced another. The pattern is now impossible to ignore. Across Oase 1 and Bacho Kiro, the story is the same. The first modern humans in Europe interbred with Neanderthals again and again, wherever the two groups met, and then most of those pioneers vanished. These people lived in a cold steppe world, hunting bison, red deer, and cave bear, and the smallest of their bones were sorted from the debris only by screening the proteins inside them, a method that can tell a human fragment from an animal one before any DNA is read. The pendants and worked bone hint at cultural exchange, or at least parallel invention, between the two kinds of human. The mainstream view credits sapiens with making the initial upper Paleolithic tools. Archaeologists picture Europe 45,000 years ago as a patchwork of overlapping, doomed groups. The speculative reading is blunt. The first Europeans were pioneers who failed. And replacement, it turns out, was not only an Ice Age story. It happened on tropical islands too, and far more recently. Number 7. The Teouma and Lapita genomes. On the island of Ifate, in the Pacific nation of Vanuatu, lies a cemetery called Teouma. It is the type site of the Lapita culture, the astonishing seafarers who first sailed into the empty islands of remote Oceania about 3,000 years ago. These were among the greatest ocean voyagers in human history. The cemetery is famous and eerie because many of the dead were buried without their skulls, which were removed after the body decomposed and sometimes replaced with symbolic objects. When ancient DNA was pulled from the earliest Teouma burials and published in 2018 in two major studies, one led by Pontus Skoglund and David Reich, one led by Cosimo Post and Johannes Krause, it broke the expected story. The first Lapita settlers derived almost all of their ancestry, roughly 95 to 99%, from an East and Southeast Asian source, a group the researchers called First Remote Oceanians. They had, at most, a few percent Papuan ancestry. Why is that a shock? Because the people living in Vanuatu today carry heavy Papuan ancestry. Everyone assumed the founders were already mixed. They were not. The founders were almost purely of Asian-related descent. And then, within just a few centuries, incoming populations with Papuan ancestry from near Oceania nearly completely replaced the Lapita gene pool. A 500-year window of DNA from the island of Malekula between about 2,500 and 2,000 years ago shows the change happening fast and unevenly. Here is the part that unsettles the tidy models. Despite this near-total genetic replacement, the Austronesian languages of the Lapita survived. The genes were swapped out. The words stayed. Language and ancestry came apart. A follow-up study in 2020, led by Mark Lipson and David Reich, resolved 3 separate phases of migration that shaped the ancestry of Vanuatu and showed the replacement was incremental, not a single invasion. It implied an enduring network of voyaging between near and remote Oceania. The result cut against both of the old models. The express train model imagined a clean, rapid Austronesian expansion straight out of Taiwan. The slow boat model imagined a gradual, heavily mixed drift eastward. The truth was neither. Genome-wide data from dozens of ancient and modern individuals, including 27 living Ni Vanuatu, showed a demographic history far messier than either idea allowed. The technical achievement here is real. Tropical heat and humidity normally destroy ancient DNA, so recovering these Pacific genomes, mostly from the dense petrous bone of the inner ear, was a triumph. The Lapita's distinctive pottery, stamped with fine dotted patterns, is the signature that first defined the culture back in the 1950s. The mainstream view describes near-total genetic turnover alongside cultural and linguistic continuity. Historians see an ongoing web of voyages rather than one clean wave. And because this touches indigenous origin stories, the results are handled with real sensitivity. Sometimes, though, the DNA does not erase a people. Sometimes it rewrites where they came from. Number 6. The Taforalt Genomes. In eastern Morocco, in a cave called the Grotte des Pigeons at Taforalt, archaeologists uncovered the remains of people who belonged to the later Stone Age Ibero-Maurusian culture. When their DNA was recovered from 7 individuals, dated between about 15,100 and 13,900 years ago, it produced a result that, for its time, seemed impossible. These are among the oldest nuclear genomes ever recovered from anywhere in Africa. The study, published in the journal Science in 2018, was led by Marieke van de Loosdrecht with Johannes Krause and Chungwon Jong. What they found scrambled every neat assumption. About 2/3 of the ancestry of the Taforalt people resembled early Holocene Near Easterners, best represented by the Natufians of the Levant. The remaining third was sub-Saharan African, related to modern West Africans and to the Hadza of Tanzania. And crucially, there was no detectable European ancestry at all, despite the culture's name, Ibero-Maurusian, which implies a link to Iberia. Take apart why this matters. The Natufian-like component predates farming. It shows a genetic connection between Africa and the Near East that existed before agriculture spread. The sub-Saharan third implies people were crossing the Sahara more than 15,000 years ago, far earlier than the later Green Sahara period, usually invoked for such travel. And the missing European ancestry destroyed the old idea that the Ibero-Maurusians came across the Strait of Gibraltar from Ice Age Spain. They did not. This was a culture whose very name told a false origin story, and only ancient DNA could expose it. The cave at Taforalt is also famous for holding one of the oldest cemeteries in the world, and for early evidence of tooth drilling and plant food processing. These were people with a settled relationship to their dead and a sophisticated use of wild plants, long before farming existed anywhere. The 7 individuals sequenced were dated tightly, between 15,100 and 13,900 years ago, which makes the mixture in their genomes all the more remarkable for its age. The cool conditions inside helped the DNA survive in a region where heat usually destroys it. The Ibero-Maurusians made tiny microlithic-backed bladelets and buried their dead with red ochre and offerings of Barbary sheep. The mainstream view now treats North Africa not as an isolated dead end, but as a crossroads of Ice Age populations. Archaeologists caution that the label Ibero-Maurusian misleads about real origins, and a genuine scientific debate continues over the direction of the gene flow, whether the Near Eastern ancestry flowed into Africa or out of it. Either way, long-distance human networks are far older than we assumed, and if a known origin story can be this wrong, what about the supposed birthplace of one of the largest language families on Earth? Number 5. Shum Laka. In the grassfields region of northwest Cameroon sits a rock shelter called Shum Laka. For decades, linguists pointed to this exact region as the likely homeland of the Bantu languages, the family spoken by hundreds of millions of people across Africa today. The site preserved human burials from about 8,000 and 3,000 years ago, spanning the transition from the Stone Age to the Metal Age. If you wanted to find the ancestors of the Bantu expansion, this seemed like the place. Ancient DNA from 4 children buried at Shum Laka was published in Nature in 2020 in a study led by Mark Lipson and David Reich at Harvard Medical School, working with the Royal Museum for Central Africa. These were among the first ancient human genomes ever recovered from West or Central Africa, a region whose hot, wet climate almost always destroys DNA. And the result overturned the assumption completely. The children did not descend from, and did not give rise to, present-day Bantu speakers. Instead, they came mainly from a previously unknown, deeply divergent lineage, distantly related to modern West Africans and to Central African hunter-gatherers. The homeland was not, genetically, the home of the people who spread its languages. Then came a detail that reached even deeper into the human past. One of the children carried the A00 Y chromosome haplogroup. This is the most divergent lineage of the human male family tree known to science. It split off from all other known male lineages roughly 200,000 to 300,000 years ago, near the very origin of our species. Its presence at Shum Laka showed that this ancient lineage had persisted in West Central Africa for at least 8,000 years. The study modeled at least 4 deeply divergent African lineages, implying that the origin of modern humans inside Africa was structured and layered, not a single ancestral stock. The people who eventually spread Bantu languages must have replaced or bypassed the earlier Shum Laka population. Recovering DNA at all in equatorial Africa required careful sampling of the dense petrous bone and heavy sequencing. The mainstream view sees a complex, deeply divided African ancestry. Archaeologists note that a linguistic homeland hid a very different genetic past. And because Bantu identity sits at the center of so much African history, the result is handled with care. It also carried a hint, a whisper of ghost populations, detectable in DNA, but almost invisible in the fossil record. Hold on to that. But first, DNA can also reveal an entire people who simply vanished from the frozen north. Number 4. The Saqqaq Genome. About 4,000 years ago, a man lived on the icy northwestern coast of Greenland. He belonged to the Saqqaq culture, the earliest known people of Greenland. We do not have his skeleton. What we have is a tuft of his hair, preserved in the permafrost at a site called Susuk, and from that single lock of hair, scientists rebuilt him. The frozen ground had acted like a vault, sealing the DNA away from heat and decay for 40 centuries. Published in Nature in February of 2010, his genome was the first ancient human genome ever sequenced to near completion. It launched an entire scientific field. The work was led by Morten Rasmussen and Eske Willerslev at the University of Copenhagen, with a team of about 52 scientists. They named him Inuk, which means man or human in Greenlandic. Hair, it turns out, can protect DNA better than bone under cold, dry Arctic conditions. From that hair, they read his biology. Inuk had brown eyes and skin darker than that of most Europeans. He had thick, dark hair and shovel-shaped front teeth. His blood type was A positive. He had dry earwax, a genetic tendency toward baldness, and a body adapted to bitter cold. He even carried the derived version of a gene called EDAR, associated with the hair and tooth traits common in East Asian populations. The team reached about 20-fold coverage of his genome, remarkable quality for 2010. But the true shock was in his family. Inuk's closest living relatives are not modern Inuit and not Native Americans. They are 3 peoples of the Siberian Far East, the Nganasan, the Koryak, and the Chukchi. This meant the Saqqaq were a separate migration wave out of Siberia, distinct from the ancestors of today's Inuit. His ancestors crossed into the Arctic roughly 5,500 years ago, and then they died out, leaving no descendants in Greenland at all. A whole population of the New World, the Paleo-Eskimos, had vanished and been replaced, and only DNA could tell that story. The Saqqaq later gave way to the Dorset people, and then to the Thule, who are the ancestors of modern Inuit. 3 successive genetically distinct peoples, one after another. The mainstream view sees multiple replacing Arctic migration waves rather than one continuous people. Archaeologists note that the cultural sequence mirrors the genetic turnover exactly, and this was one of the first ethical flashpoints in the field, the question of sequencing an identifiable ancient individual. Think about it. A single strand of hair rewrote the history of the Arctic, and a single broken skull was about to rewrite the timeline of Europe. Number 3. Zlatý kůň. On a hill in Czechia called Zlatý kůň, which means golden horse, workers in the 1950s found a fragmented human skull inside a cave system. The skull belonged to a woman. It had been broken, and some of the fragments had even been gnawed by a hyena. For years, no one could pin down how old she was. Radiocarbon dating kept failing because the bone was contaminated. Her true age would eventually be revealed not by chemistry, but by her own DNA. Her genome was published in Nature Ecology and Evolution in 2021, in a study led by Kay Prüfer and Johannes Krause, and it earned her a record. Zlatý Kun is the oldest reconstructed modern human genome to date, more than 45,000 years old, roughly the same age as, or slightly older than, Ust-Ishim man from Siberia. The way they dated her is the elegant heart of this story. Instead of relying on radiocarbon, they measured the length of the Neanderthal segments in her genome. Remember the molecular clock. Every generation, recombination breaks inherited Neanderthal DNA into shorter pieces, so the length of those segments counts the generations since the interbreeding event. Her Neanderthal segments were longer than those of any modern human sequenced before her, even longer than Ust-Ishim's. That meant she lived very soon after the Great Neanderthal Admixture Pulse. Her genome carries about 3% Neanderthal ancestry, similar to other Upper Paleolithic hunter-gatherers. And once again, the ending is the same. This woman belonged to a population that contributed genetically to neither later Europeans nor later Asians. Her entire lineage disappeared. She sits alongside Oase 1 and the Bacho Kiro people as another founder who failed. Her DNA places her near the very base of the split between Eurasian lineages, and it suggests that modern humans reached the heart of Europe earlier than the DNA of living Europeans would imply. Think about the journey of this skull, discovered in the 1950s, and only decoded 70 years later when the right method arrived. It helped calibrate the timing of the main Neanderthal admixture to roughly 45,000 to 50,000 years ago. The hyena damage and the fragmentation made contamination control especially hard. The mainstream view calls her an early European wave that went extinct. Archaeologists see repeated failed colonizations before the successful peopling of the continent. A few researchers still debate whether her real age is closer to 43,000 years. But extinct human DNA did not only vanish into these dead ends. Some of it survived. Some of it is keeping people alive right now. Number 2. The Tibetan EPS1 Denisovan altitude gene. Return to the roof of the world, the Tibetan Plateau, where the Xiahe jaw began this list. Living Tibetans do something that should not be possible. They thrive at altitudes that leave lowlanders gasping, dizzy, and sick. The secret sits in a single gene called EPAS1, part of the body's response to low oxygen. And that gene shows the most extreme signature of natural selection found in any human population on Earth. When lowlanders go to high altitude, their bodies overproduce red blood cells, thickening the blood dangerously and causing chronic mountain sickness. Hemoglobin, the molecule that carries oxygen, climbs to harmful levels. The Tibetan version of EPAS1 prevents that. It tunes the oxygen response so the blood stays efficient without becoming thick and dangerous. And in 2014, a study in Nature, led by Emilia Huerta Sanchez and Rasmus Nielsen, revealed where that life-saving variant came from. It did not evolve from scratch. It was inherited from the Denisovans. The researchers resequenced the EPS1 region in 40 Tibetans and 40 Han Chinese. They found a highly distinctive stretch of DNA, a haplotype about 32.7 kilobases long. It was common in Tibetans, rare in Han Chinese, and genetically closer to the Denisovan sequence than to any other modern population. This is the textbook case of what scientists call adaptive introgression, DNA from an extinct human species that provides a real survival advantage in living people. And it is not rare. This Denisovan-derived haplotype occurs at roughly 80 to 87% frequency among Tibetan chromosomes. Now, connect the threads. The Xiahe Denisovan lived at 3,280 meters, high on the plateau, more than 100,000 years ago. The Denisovans likely already carried an altitude adaptation. When modern humans reached the plateau and interbred with them, they inherited a ready-made solution to thin air, a shortcut that would have taken tens of thousands of years to evolve on its own. A follow-up study in 2021 in the journal PNAS traced the history of this Denisovan haplotype across Himalayan populations. Sit with what this means. A piece of the genome of an extinct human species is doing essential, life-sustaining work inside living bodies every single day. Han Chinese, who split from Tibetans relatively recently, mostly lack the variant, which sharpens the contrast. The 32.7 kilo base block is unusually long and intact, a hallmark of strong, recent selection. Denisovan DNA also shows up in immune genes and in Melanesian and East Asian populations, but EPAS1 is the clearest adaptive case of all. It reframes archaic DNA not as junk baggage, but as a toolkit that some populations actively kept. The mainstream accepts adaptive introgression as real and beneficial. This story is often downplayed because the idea that extinct human DNA keeps us alive unsettles a clean origin story. But if we can inherit genes from an extinct species we have actually found, what about one we have never found at all? Number 1. The West African Ghost Archaic Lineage. Here's the find that sits at the very top of this list, because it asks us to accept the existence of a whole human species that no one has ever seen. In 2020, a study appeared in the journal Science Advances by Aaron Durvasula and Sriram Sankararaman of the University of California, Los Angeles. Its title was Quiet. Its implications were not. They reported that 4 West African populations carry DNA from an archaic human lineage that has never been identified from a single fossil. There is no bone. There is no tooth. There is no skull. And so the source is called a ghost population because it exists only as a pattern buried in living DNA. The signal appears strongly in the Yoruba and the Mende peoples of West Africa. These populations derive an estimated 2 to 19% of their ancestry from this unknown group. Think about that range. Up to nearly a fifth of some people's ancestry may come from a species science cannot name. The details make it stranger. The ghost lineage split from the ancestors of modern humans and Neanderthals somewhere between about 360,000 and 1,020,000 years ago. That is deep. Deeper than the split with Neanderthals themselves. And the interbreeding with the ancestors of present-day West Africans happened within the last 124,000 years, possibly much more recently. To find it, the researchers used a clever method that can detect archaic ancestry without needing a reference genome from the archaic species itself, which is essential when you have no fossil to compare against. They found 33 places in the genome where the archaic segment frequency exceeds 50% in the Yoruba, and 37 such places in the Mende. High-frequency segments like that hint that some of this ghost DNA may have been favored by natural selection, kept because it was useful. This parallels the Neanderthal and Denisovan interbreeding outside Africa, but it involves a completely unknown African hominin. And it overturns the old, comfortable idea that interbreeding with archaic humans only happened after modern humans left Africa. It happened at home, too. Why has no one found the bones? Because Africa's fossil record is sparse, and its climate destroys DNA, so this entire species may be effectively invisible except in our genomes. There are candidate faces, archaic-looking African fossils like the remains from Iwo Eleru in Nigeria, but none is a confirmed match. The finding implies that our species did not arise from a single pure ancestral group, but from a structured web of interbreeding African populations. It fits the growing shift away from a simple out-of-one-place origin toward a pan-African, many-population model, and independent studies have found similar deep archaic signals in other African groups. The debate is honest and ongoing. The mainstream sees genuine ghost introgression from an unknown archaic hominin. Archaeologists hope candidate fossils may one day match the signal, and some caution that complex ancient population structure could, in principle, mimic an introgression signal. But whichever way it resolves, the core fact remains staggering. Millions of living people carry pieces of a human species that we can measure but cannot see, an ancestor still hiding in plain sight, written into the blood. Step back, and a single thread runs through the oldest finds on this list. Oase 1, the Bacho Kiro people, and the woman from Zlatý all share the same fate. They were among the very first modern humans in Europe. They interbred with Neanderthals, sometimes only a handful of generations before they lived. And then they vanished, leaving no genetic trace in anyone alive today. The peopling of Europe was not a single triumphant march out of Africa. It was a series of failed colonizations, waves of pioneers who arrived, mixed, and died out, one after another. The tool that revealed all of this is the same in every case, the molecular clock hidden in the length of Neanderthal DNA segments. Long segments meant recent mixing. Short segments meant ancient mixing. That ruler let scientists read family history from a genome. And notice the irony. The Neanderthal DNA that modern Europeans carry today did not come from these early, high Neanderthal pioneers. It came from a different, earlier, successful admixture event. The losers of prehistory left nothing behind. The Teouma story from the Pacific taught the same lesson in a warmer place and a nearer time. Founders can be erased fast, within centuries, even as their language survives. Picture the scale of it. A small band of pioneers pushes into a cold, unfamiliar continent already occupied by Neanderthals. They survive for a while. They have children with the people they meet. And then, over a few centuries, disease, competition, bad luck, or a shrinking population simply ends the line. It happened at Oase, in Romania. It happened at Bacho Kiro, in Bulgaria. It happened at Zlatý, in Czechia. 3 separate places, 3 separate failures, all reading the same. The mainstream frames this as demographic swamping and the extinction of small populations. Archaeologists picture overlapping frontier groups meeting at contact zones. The more speculative voices suspect that many more failed hybrid waves are still buried, waiting. And there is a reason this story stays quiet. A history of failure resists the tidy, comforting origin narrative that audiences expect. Extinction, not smooth continuity, is the true theme of the deep human record. Which raises the obvious question. If the losers vanished, what exactly did the winners keep from the archaic humans they met? The answer flips the whole story around. Some archaic DNA was not lost, and it was not junk. It was kept deliberately by natural selection because it helped people survive. EPAS-1 is the clearest example. Denisovan DNA lets Tibetans live and breathe in air so thin it would sicken almost anyone else. A gene from an extinct species is at work in living lungs and living blood. It does not stop there. Archaic-derived immune genes and metabolic genes persist in many populations around the world. Neanderthal variants influence immunity, skin, metabolism, and even the body's response to day length and sleep. The Xiahe jaw gave this idea a physical face, a Denisovan living at more than 3,000 meters, carrying the very adaptation that would one day flow into modern Highlanders. The logic is simple and profound. Selection acts like a filter. Harmful stretches of archaic DNA were purged from our genomes over generations. Useful stretches were retained and even spread. Interbreeding became a shortcut, a way to acquire adaptations that would otherwise take vast stretches of time to evolve from scratch. Evolution, in effect, recycled the best tools of a lost species into us. Consider the learning curve this represents. To evolve a brand new adaptation to thin air, a population would need countless generations of mutation and selection, with many dying along the way. Interbreeding skipped all of it. The Denisovans had already paid that price over their own long history on the plateau, and modern humans simply inherited the finished result in a single act of mixing. It is one of the fastest routes to adaptation that biology allows. The mainstream now treats adaptive introgression as a major evolutionary force, not a curiosity. Archaeologists increasingly see contact zones between species as engines of adaptation, not only of conflict, and the speculative edge suspects that unknown archaic genes may still shape traits we have not yet mapped. This is a hard idea to sit with, the recognition that we are part extinct human by design, stitched together from lineages that no longer exist. And the strangest donor of all is one we have never even found. The West African ghost lineage is the purest example of a new and unsettling kind of discovery. It is a human ancestor inferred entirely from DNA, with no fossil to anchor it. And it is not alone. The deeply divergent Aor-0 lineage found at Shum Laka hints at the same hidden depth in the African past. Ancient branches of humanity that barely register in the fossil record. The origin of our own species, Homo sapiens, is starting to look less like a single trunk and more like a braided river, many channels splitting and rejoining across the African continent. Ghost populations, groups we can detect statistically but cannot yet see, are becoming a standard concept in human genetics. DNA can now prove that a species existed without anyone ever finding its bones. Why is the African record so thin? Not because the past was simple, but because the climate is hostile to both fossils and DNA, and because far less of Africa has been intensively excavated. The absence of bones is a gap in our searching, not a gap in the story. The multi-regional within Africa model, once fringe, is steadily gaining ground on the strength of results like these. There is a strange inversion at work here. For most of the 20th century, fossils led and genetics followed. A bone was found, described, and slotted into the family tree. Now the order can reverse. The genetics can announce that a population existed, describe roughly when it split off and when it interbred, and then send the fossil hunters out to look for a body that may or may not ever turn up. The West African ghost may already be sitting, unrecognized, in a museum drawer somewhere, its bones cataloged, but its true identity unread. The mainstream accepts genuine archaic introgression inside Africa. Archaeologists hold out hope that candidate fossils will eventually match the genetic signals. And a cautious minority argues that ancient population structure could produce a similar pattern without a distinct ghost species. The methods here, the ability to detect an archaic ancestor with no reference genome at all, are as important as the discoveries themselves. Which leads to a fair question. Why do so many of these landmark results stay out of the spotlight? The honest answer is not conspiracy. It is caution, complexity, and consequence. These finds live at the edge of what the methods can do. Paleoproteomics and reference-free archaic detection are young techniques, and extraordinary claims built on them invite careful skepticism. Degraded DNA and contamination make many of these results hard-won and genuinely contestable. There are ethical stakes too. Sequencing an identifiable ancient individual, as with the Saqqaq man, raises real questions about consent, descent, and repatriation. Many results touch living identities directly. The genetics of Bantu origins, of Pacific settlement, of deep African ancestry are sensitive precisely because they matter to real communities. The finding that language and genes came apart in Vanuatu unsettles origin narratives that people hold dear, and the shape of these stories works against them. Failed founders and unseen ghost species do not fit the comforting, triumphant arc that popular science usually prefers. Single specimens and small sample sizes limit how far any conclusion can be pushed. Competing statistical models can read the same data in different ways. Science communication struggles to convey uncertainty without sounding like doubt. There is also the simple problem of numbers. A single jaw, a single tuft of hair, a single broken skull. Each of these landmark finds often rests on just one individual, or a handful. One genome can overturn a textbook, but it cannot, by itself, describe an entire vanished people. Scientists know this, so they hedge, and the hedging reads to the public as doubt, even when the core result is solid. The mainstream stance is that these results are robust but appropriately hedged. Archaeologists insist that fossils and DNA must be reconciled, not ranked against each other. A fringe voice claims that mainstream science suppresses inconvenient ancestry, but that charge is almost always overstated. The real picture is quieter and more human. These finds stay in the background, not because they are hidden, but because they are careful. Step back one more time, and the 10 discoveries tell a single story about who we are. Every find on this list shows the same thing. DNA has rewritten a story that bones alone could never tell. We are the product of interbreeding, of extinction, of replacement, and of inheritance. The people who arrived first in a place are very often not the ancestors of those who live there now. Pioneers failed. Whole peoples vanished, and other lineages quietly persisted for hundreds of thousands of years. Extinct species live on inside us, and sometimes they keep us alive, breath by breath, in the thin air of the highest plateaus on Earth. Some of our ancestors exist only as patterns in living genomes, never as a skull in a museum drawer. The human family tree is not a clean line running from apes to us. It is a tangled, braided river, full of dead ends and hidden channels, and species we are only beginning to sense. Ancient DNA has made prehistory more honest and far stranger than the version in old textbooks. These discoveries are not concealed by conspiracy. They are muted by complexity, by caution, and by the sheer difficulty of the science. But they are real, and they are inside us. We carry the dead with us, the failed founders, the lost peoples of the Arctic, and the unseen species of Africa whose bones we may never hold. Our deepest history is not written only in stone, or in fossils, or in the artifacts our ancestors left behind. It is written in the blood of the living, in every cell, waiting for the next method, the next discovery, to read it aloud.